Okay. Hello, everybody. Thank you. And thank you, CPATH, for organizing this. I've learned a lot today. We all have in my group, and it's really a pleasure to be here. I took an approach to this. I understood we are case studies, and so I'm actually going to share our journey, actually engaging with the FDA in our first DDT for qualification. We are a barrier-focused group. The outline of the 5 DDTs that we proposed in the in our actual application to the TRACE program are shown there. The first one is a model of a bone infection, and that's actually the one that we're advancing because of its simplicity and because of also our experience doing some actual drug discovery using that platform. The other models that you see there are all examples of basically an immune system in a vascular interface interacting with tissue. There's a multiple sclerosis model where we're looking at T-cell invasion, a human tendon on a chip, which is a fibrosis model, a sepsis model, and then a model, an immune disorder known as juvenile dermatomyositis coming out of George Trusky's lab. So this is our team at the highest level. These are the other co-PIs, Jonah Dama, who's here, who's a regulatory scientist in the group. And George Trusky is also here. He's a lead at Duke. We have Hani Awad and Ben Miller also as co-PIs at the University of Rochester. This is the ambitious Gantt chart that we put together for ourselves. We decided we were going to go ahead and do 5 DDTs, particularly because the first one actually is so simple that it may not qualify, it probably shouldn't qualify as classic MPS, but it is an assay for drug discovery, and we thought it worthy of qualification, so we put it first. And then you see the others actually basically in order of complexity. So we started with the simplest one and we tried to move towards the more complex ones. We are on track except for this business of engaging the FDA and how quickly they are engaging us back. So we are for the first one scheduled to submit a letter of intent. We've drafted a letter of intent, and we are actually in the process of engaging the FDA for a pre-letter of intent. So this is kind of going a bit slower than we anticipated, but we are pretty much in terms of the development of the tool and the SOPs in the interlaboratory testing, we're on track with all of that. We also have some interest from pharma, which is notable because as we engage pharma, which is part of our mission, we're actually getting interests that are outside of the DDTs or that we had actually proposed in the proposal. So it's actually bringing on additional opportunities, but also additional resources are required. So I'm going to actually take you through the letter of intent development process that we've been going through. So this is really meant to be very granular, hopefully helpful to some people when you see our own mistakes and the ways in which we were naive. I also hope it's helpful to us and that there will be folks out there that can help us move through. So we're going to talk about the letter of intent, not just the context of use, because we've sort of found that working on the letter of intent is the best way to think about the context of use. I need to tell you first about the disease context. So the DDT that we're advancing is about osteomyelitis. So this is a deep bone infection. The bone has a small canal system known as the osteocyte lacunar canalicular network, and Staph aureus finds its way into these cracks in bone. And once it's there, the immune system can't get to it, nor can drugs. So it's absolutely devastating. And it's actually quite surprising that they can do this. People who have studied Staph aureus only know it to be a round coccus of about a micron in size, or a little bit bigger than a micron in size. And in no instances has it ever been seen to be elongated, except for in these small cracks of bone. So we were asked as part of the original discovery of the fact that Staph aureus could do this to develop a simple system to show that Staph aureus has this capacity to change into a non-round shape and squeeze through small confines. And we did this by making a simple transwell-like system in which the pores were all the same size and all smaller than Staph aureus has ever been seen to be. And you can see the data sort of validating all that. And lo and behold, the bacteria do pop through by mechanisms that's still not quite understood and occupy the other side. What we did then is we took this into screening so that we have used these tools as screens, first genetic screens. So we took libraries of Staph aureus and identified the genes that were essential. We also discredited a number of hypotheses about which genes in Staph aureus would be essential with the same tool. So these were predictive of the eventual mouse models, the chip itself, the transmission through the pores of the chip were actually predictive of whether or not the infection happened in mice. And then we actually took this to the tail end as a functional screen of a drug development pipeline. A whole lot of other types of screens were done first, and at the tail end as a functional assay, this was used to identify the best compounds. So a lead identification, which is obviously not what we're supposed to be qualifying, but certainly this from a pharmaceutical development point of view is a drug development tool. So this is what we're going into the qualification process with. And what I want to sort of highlight here are three areas of the letter of intent that we found to be most useful and actually co-developing them to help us think about this. The drug development need statement, the context of use statement, and the drug development use statement. There are instructions here in this template, and reading those instructions and then trying to write to all three at the same time is how we've approached this. So I'm going to first show you our first attempt, which looks like this. This basically says this osteomyelitis is terrible. If you end up with this disease, for which there is no actual cure, you live with a lifelong risk of reinfection, sepsis, and amputation. So it's a terrible thing. It's rare, but it does happen. But really all this development need statement is that we need new drugs and we don't have them. So there's a need for the development of drugs, but that is not sufficient. We've come to clearly understand for qualification. The context of use statement, this is going all the way back to our original one in our RFA, which is good enough for NIH reviewers, but not good enough for the FDA. Actually, kind of tells you that our mindset was still sort of in the drug development pipeline from the pharmaceutical point of view, which is a pitfall for efficacy models in particular that are going into the ISTAN program. We actually said our context of use was for prediction of drugs that will block this colonization of the cortical network and bone. This is what our drug development use LOI look like. This is the drug development statement look like. And basically, we did start with the lead compound being identified, but because the call called for a flow chart of some kind, we actually built in this sort of decision, which ends up looking very much like a internal decision in a company. So we made some early mistakes. We had a meeting recently, thanks to this wonderful program. We actually have pre-LOI discussions with the FDA and with CPATH, and we had this presented this type of development to them, and we got the feedback that you've heard from York, which is, looks like this might be for an internal company decision. So this is the pitfall, I think, that many MPS going into the ISTAM program have fallen through. I followed up immediately with Graham Marsh, who was also on the call, and it just highlighted an issue here, which was that the animal models are extremely limited. They cannot actually do the efficacy assessment. They can never address the actual mechanism of action. And Graham responded, making the case that a current preclinical model has a deficit that your system addresses, make a strong argument for qualification as a DDT. So even if the farm user still uses the animal test, you've made a tool available as part of a weight of evidence assessment because there's an aspect of the biology that the animal doesn't address. So this was the light bulb that turned our statements into statements that really hone in on the fact that the only way a drug developer is going to assess whether or not the mechanism of action is actually working for their drug and use that as part of an IND package was with our tool. You can't actually do it with the animals. You can't do it with any other in vitro assay. So in statement 3, you'll see language now that says this is the only preclinical tool that simulates a submicron architecture of the OLCN, making the chip indispensable for assessing the specificity of drugs. The context of use also has this type of language in it now. I'm still not sure after seeing the emulate example today that this is still totally right, but we are refining and refining to emphasize the fact that there is no other way to get at the mechanism of action other than using our tool. And then finally, I think some of the language that shows up in 6 actually looks like it should be in the context of use. We anticipate the development of drug development programs that are targeting the immune evasion mechanism used by Staph aureus. It will be the only reliable preclinical assay to demonstrate the effectiveness of drug in hindering the deformation elongation mechanisms of Staph aureus. We believe this is absolutely true. There's no other way to show that a drug is actually targeting this very specialized pathway, which otherwise only exists inside of bone. So this is where we're at. We still don't have a qualified, or we don't even have an invitation to the letter of intent process, but we are drafting this and really working these 3 and getting as much feedback as possible. And in fact, if there's anybody in the room who wants to offer more, we're all ears. So thanks.